Thank you for joining this conversation between two physician scientists and longtime colleagues, each of us with a career-long focus on lupus research. I'm Peggy Crow. I'm until very recently was the physician-in-chief at Hospital for Special Surgery, and I'm a professor of medicine at Weill Cornell Medical College. I'm a rheumatologist, but a longtime lupus researcher. Very pleased to now be back in my lab after completing my time as physician-in-chief. And Jane?
I'm Jane Salmon. I'm the director of the Lupus and APS Center at Hospital for Special Surgery, and I'm professor of medicine at Weill Cornell Medicine and professor of medicine and obstetrics and gynecology at Weill Cornell, and you'll understand why that appointment is important as we continue our conversation. And I'm also Dean for Faculty Affairs at Weill Cornell because I'm very interested in mentorship and faculty and advancement. But the bulk of my focus over more years than I want to admit has been studying mediators and mechanisms of tissue damage in lupus at the Hospital for Special Surgery.
I'm going to start by asking Jane, just tell me, how would you describe your area of research, your research focus?
I'm motivated and driven by the challenges that patients face with lupus and end organ disease as opposed to having autoimmunity. Autoimmunity doesn't really bother them. End organ disease is what causes decreased length and quality of life. So I've studied kidney disease, I've studied cardiovascular disease, and my focus in the last 20 years has been understanding pregnancy complications in women with lupus. So that's really what drives me. I look at mice and mouse models that can recapitulate the problems that patients have, try to prevent them in the mice, then try to understand the prevalence and the predictors of different phenotypes in patients, like who's going to get atherosclerosis and when will they get it? What are the clues to prevent renal disease? What are the genetic risk factors? Similarly, in pregnancy, what are the clinical and biomarkers that identify women at risk? And then once we know who's going to get the problem and the mice tell us what the intervention is, the dream is to do a trial and try to prevent the problem in the patients. So that's how I've approached lupus investigation.
What was most important in defining this area of research for you?
I think I started medical school as one of very few women in my class, but it didn't feel strange. I wanted to do what I did, and I just went ahead with blinders, and it didn't bother me that I was a girl and they were guys. I just did what I had to do, and I did it reasonably well. But then when I started to interact with patients, it became clear that the diseases of interest, we're talking the late 70s, mid-70s, the diseases of interest were really diseases of men. And women weren't in trials. And I met lupus patients, and they were all young women. And the difference between the patients and me was that they were sick and I wasn't. And no one really cared. I mean, it was a curious disease. It was a rare disease. And I was always interested in science. I was in the MSTP program at Columbia. I was actually the first woman in that program, and I didn't even notice that I was the only woman in that program. And I did my lab work, and I was curious. And I said, this is an unmet need. This is something I can identify with. The patients excited me. The disease was a mystery. So that's really what drove it. And then moving forward, when I was more clinically active in training and earlier in my career, you talk to the patients, and what mattered to them is what drove the laboratory question.
So when you tackle a research problem, like trying to achieve better pregnancy outcomes for patients, how do you structure the project? How do you actually implement it and get it done?
So there's rigor, there's organization, and there's serendipity. And so I guess I migrated towards the pregnancy studies because I was always interested in antibody-mediated injury. And patients with APS have thrombosis, and that was a thrombophilic disease. And what did antibodies have to do with it except there were markers? So I set up a mouse model of antibody-mediated pregnancy complications. You take a pregnant mouse, inject APLs, and they get growth restriction and they get fetal loss, just like people do. And then the question was, what drove it? How did the antibody screw up placental development? And I was interested in neutrophils, monocytes, FC receptors, but not complement. I mean, complement was something I was totally not interested in. And serendipity, there was a paper that got published that said that complement inhibition was required for normal pregnancy. So if you knock out complement inhibitory molecules in a mouse, the mouse has a lot of miscarriages and a lot of inflammation. And at that point, I got a phone call from a colleague I had met at an ACR meeting on a committee who was a complement biologist, and we discussed this paper. And he said, I have this molecule that inhibits complement activation. Why don't you put it in your mouse model? And I kind of said, it's not really a good idea. Complement has nothing to do with any of this stuff. And I did it. And it rescued all the mice pregnancies, which was astounding and dogma challenging. So that was serendipity. I had the model going. And then I spent the next, 5 years looking at in a rigorous way, looking at the role of every component of complement and every complement receptor to define it. So you're asking how you do it. It was very methodical. We got knockout mice, we got compounds, we got monoclonal antibody blockers, and the experiments were very simple. You block the pathway in the pregnant mouse that gets APL, and you look at the outcome. So it's kind of brute force in a certain sense. And then the human studies, you have to make a team. So I became a cheerleader. I brought together people who were interested in lupus pregnancy and said, let's enroll patients and figure out who gets in trouble and who doesn't, because we really haven't a clue how to counsel patients. Mm-hmm. And that took another 10 years.
I know very well, because I work close to you, that you don't actually go inject the mice yourself, and you don't kill the mice, and you don't pipette under the hood. No. So how do you bring together and motivate the people who work with you to be sure that they're doing things well and that they get it done and that things move forward?
It has a lot to do with trust. They're also excited about the possibility of translating a basic observation to patients. Even the PhD scientists want to apply what they learn to patients. So I think that's fantastically motivating. And it's very unusual to be able to almost realistically aspire to do that, because there's such a big gap between what we do in the lab and the application. But the gap's really small in my area of investigation. That's because I can't tolerate big gaps. So people who work with me, I think somehow are excited, or I pick them right, or I motivate them. But you're right, I do create teams. I collaborate with people all over the country. They send me reagents. I'm not sure what it is. I think also I'm generous about authorship and about concepts, and really give credit to people who gave me ideas. And I do cross-disciplinary work, and I guess because it's fairly broad, you have to have a team. You're in many ways the opposite. You're tall, you're blonde, I'm short, I have dark hair. You focus in a very specific area. You're bold in your inquiry. And I want to understand how you got this boldness. You get these very deep thoughts about the immune system and about the genome and about viruses. They're really complicated and they're fundamental. They're different from what I do. And you get excited by it. How are you comfortable taking those risks? I talked about being uncomfortable with gaps.
Well, you're right. We're very different. And I won't go so far as to say that I'm not driven by patients and their diseases and their bad outcomes, because of course I care about that, but it's really not at all what drives me. I think I'm very curious and respond to very complex problems and puzzles. And somewhere along the way, I found lupus, and there's really no disease or, medical scenario that I think is as challenging. It's based in the immune system, and I learned even before medical school a bit about the immune system and recognized it as this interacting network of molecules and cells, and they work well together, or they fight each other. And it's just this tremendous, challenging puzzle to me. And lupus is the model for having a disease that allows me to think about all this together. And it's become even more complicated because it's not just the immune system, as you're suggesting. Really, it's our kind of genetic endowment, how we differ from one person to another. It's our interaction with the environment and viruses. So I think I respond to complexity. We all develop a narrative of our lives. And I think that narrative is very much grounded in our family, how we grew up, and, at a very early age, how we responded to all that. And then we also are, genetically wired in different ways. And I think I must be wired in a way that I love to bring together information and try to connect the dots in some kind of meaningful way, which is very difficult to do, but it's challenging. And just a bit of an anecdote goes back to when I was in an immunology lab before medical school. And this was in, say, the mid-1970s. And I came across a book by a woman immunologist who happened to be working at Sloan Kettering at the time. Her name was Maria de Sousa. And it was about lymphocytes and how they move from lymph nodes to the blood and to the lymph and how they interact each other. And it was during the era when there was just getting to be an understanding of how antigen-presenting cells worked, or that there could be suppressing cells and that sort of thing. And I read her book, and I think she might have drawn the analogy to a complex family. And it really resonated with me. And ever since, I've thought that studying the immune system in the setting of a very complex disease maybe is a way to work out my own narrative, relevant to whatever was meaningful to me early on in understanding the complexities of human interactions, but beyond that even, kind of interactions among all living things and our genomes and our host responses. And so I'm just very, driven to figure stuff out.
That's a fantastic analogy, and it gives me a whole new insight in you and why you do what you do. But how do you rein it in? Because it's curious, but you have to do an experiment.
Right. So it can be I have difficulty reining it in sometimes. What I've done or organized in the lab, with obviously help from others, is to collect in a very detailed way data from patients over time, and then generated biologic data on those patients. So that data kind of forms my raw material for trying to pull together themes and signals and messages. And, you were asking me recently, how did I come up with the idea of type 1 interferon in lupus? And I thought about that. And it's not that I came up with it, the data came up with it. And, maybe the way I address the data a little bit reflects what I do. I mean, we had data from patients. It was gene expression data that at the time was kind of a relatively new kind of data. The bioinformatic tools that were available, at least to me at that time, were extremely unsophisticated, and I really didn't use them. And what I did was I took every single one of these gene transcripts that was shown to be differentially expressed between lupus patients and healthy people, and I read everything I could read about it. And what emerged from that was, the vast majority of these differentially expressed gene transcripts seem to be related to interferon, or what interferon does to the immune system. So the observation arose out of the data, and then that takes me to the next step. Well, then, how do we understand this? And I think then another observation, or another example of how I guess my head works, is that we carefully related interferon activation to clinical and laboratory data. And what jumped out there was not anything I would have predicted. It was the fact that there was an association with autoantibodies that target RNA-containing particles. And so then you follow the thread of RNA, and what does that mean, and how does that work? So I think it starts with, that raw material data that's very much patient-derived, but then using that and trying to draw messages from it. But then I think the other aspect that's very, very important is the literature, and kind of sometimes odd parts of the literature that are not obviously even related to medicine, and trying to make connections. So that's where I think, we're all wired differently. And I think there's something about my brain that likes to pull in these threads and weave them together. And it can be very difficult. I mean, it can be kind of exhausting because it's hard, but then sometimes things will just snap into place and I'll feel like, I get it now. And then I can move on to the next step.
It's somewhat agnostic of machine learning in a way, because you have all this data and you're not coming to it with a fundamental bias. Right. You're coming to it as though you want to discover what it's telling you, as opposed to what many people do is push the data to tell them what they want to hear, or to see if the data is telling them what they want to hear.
So how do you view the role of mentors to you during the course of your career? And then secondly, how do you approach mentoring, young rheumatologists, physician scientists in the early stage of their development?
I guess, I think about mentoring a lot because I'm invested in it at Cornell, and because of my wish to, succession planning, and to replace myself and my peers, to find people to do what I do and love what I do. I didn't really have mentors. No one looked like me when I was training. It didn't bother me, but I found that even in studying lupus, HSS was a place where lupus was really the favored disease for research. And historically, fantastic things came out of lupus research at HSS. So it was a fertile area for me to study. There were a lot of patients. There was a lot of knowledge about the disease and the effector mechanisms, which is what I focused on. But I didn't have a particular mentor who I modeled either my career or myself after. But I've mentored a lot of people, maybe in response to this sense of longing for a maternal figure to mentor me. So I don't think I coddle my mentees, but I pick them carefully. And I rarely mentor men. I mentor women predominantly, and that's just kind of how it happens. So what I've tried to do is show my mentees you can be a passionate scientist and you can have a life. a family life or a cultural life. And I think I have a three-dimensionality, and I try to mentor them in a way that basically my advice is find what you're passionate about. Look really hard for that and run with it. But don't abandon everything else, and recognize that there are a lot of bumps in the road. I always tell them that if all their experiments work, they're not taking enough risks. And somehow it becomes a sort of a girl's family. It may also be that because I have two sons and a husband, I lived in an all male dominant environment. I trained in a male dominant career. I work in a male dominant hospital, which is changing a little. That surrounding myself with these X chromosomes, I think, felt really good and feels good for me. I mean, I don't know why. It just feels good, and I'm so proud of the people I've trained, even though most of them end up being very different from me in what they want to do. I feel like they've been empowered so that they can do whatever they want, and I think I'm lucky. I don't know how it was that I was empowered to do what I wanted to do. I'm not really sure why, but I try to convey that.
Well, for me, I'm thinking about, my mentors, and maybe more than you. I feel like I did have mentors, but they were different from each other. The person who headed my lab that I worked in before medical school was amazing in giving me opportunities to almost act like I had some independence, which was ridiculous at the time because I didn't know anything. But I guess he gave me the sense that I could have ideas and could even implement them, through experiments and so on. And he was also very directive to me because he basically told me, you've got to go to medical school. And I was not at all, it was a possibility that it occurred to me, but it was not at all my plan. In fact, I had crazy possible plans back then. I interviewed as a paralegal. I think I applied for a job at an insurance company. Crazy stuff, not medicine. So I do think that here was somebody who was looking out for me and trying to help me go in a good direction that would be a good fit for me. So that was one kind of mentoring, and that was just pure good luck that I happened to encounter him. And then, I ended up in Henry Kunkel's lab at Rockefeller after residency. And he was quite well known, I think, for his mentorship style, which was experienced, I think, by many people as incredibly stressful and difficult. And really what it was he was kind of the father figure, and you either pleased him by working incredibly hard and coming up with very interesting observations and just being smart and knowledgeable and just, overall good, or you failed. And all of that was almost without any direction from him. I've told people that the way he would give guidance would be to toss down, a Xerox copy of a publication that had just come out on your desk and say, you might find this a little interesting. And what that meant was, you better work on something really, like this, either repeat it or take off from it. So it was incredibly a light touch in terms of giving guidance, feedback. I would show him my data and he might just nod. That's about as much direction as I would get. But I guess I responded to that. And maybe it also, in anyone who can thrive in that kind of setting, stimulates you to maybe be more creative because you're very much on your own. It's, so much of what you're doing is coming from yourself but, with either negative or positive reinforcement from him. So he, I would say he was my most influential role model, but then I think he's, in a way, my model for how I might mentor others. Mm-hmm. And, I do think I have a less directive approach than you do. And, part of that is, I think of myself as shy. I enjoy having people in my lab and I want them to be successful, but I don't want to push them in a direction. So I might try my best to tell them what I'm thinking, tell them what I've heard about, what I've read. And I might say, you might think about trying that. And now that I'm saying it, that's so much reminiscent of the way Henry Kunkel would speak to me. So, I think we model ourselves on others. And then I think that style of modeling also has to resonate with our own personalities or temperaments.
No, I think our differences are hardwired. Just the way you thought you were going to do something different than what you're doing. I always wanted to be a scientist. I didn't want to go to medical school. I wanted to be a scientist, but there was tremendous family pressure to be sure that I was employable. So they said, you can be a scientist after you go to medical school. And I was fortunate to be able to be an MSTP student because otherwise there would have been a family war.
Yeah. In my case, I was doing something completely different than anything my family had ever experienced. No one, well, I have a distant cousin who's a physician, but that's about it. So in a way, it was taking this road in spite of what my parents or my family thought I might do.
Well, I sort of did that too. They wanted me to see patients. So I went to the lab, I think, as a rebellion. It's what I wanted to do, but it was also somewhat rebellious, but it made me happy and it was exciting.
Yeah. I think rebellion is a characteristic we all have, or kind of anger and, doing things in spite of others. And, hopefully those sorts of drivers are channeled in productive directions. But everybody has very complex, I think, motivation.
Writers and musicians and dancers, they're fighting something and they're just channeling it in a fantastic way that people benefit. Perhaps not the people they're rebelling against, but community and society. I think, everyone's angry about something these days, more so perhaps. So
let's talk about what's going on in lupus research now, and what do we think is particularly either exciting or promising, or maybe exciting and promising.
So I'll couch it in clinical terms. And I think the key discovery that's going to be made is matching, or should be made, is trying to match patients with drugs so that we have a better sense of whom to treat with what intervention. Both the genetic, the gene expression, the epigenetic explosion in data, I hope, is going to tell us some of that. So I see that as the fundamental change or potential in lupus therapeutics. And that has to be married with all the new targets that we have, and all the new successful or semi-successful interventions that we're embracing right now, and are either starting, going to start, or finishing. So that's where I see it, is figuring out who's going to respond to what. Because people like you study initiating mechanisms, and if we understand the initiators, we can block them. But I think similarly, drugs that block effector mechanisms, you can still have autoimmunity and not have kidney disease, are also going to play a role. And the targets are different, so the side effect profiles will be different.
So I'm thinking, what do I think is most exciting? I do think that the development of very sophisticated analytic tools, machine learning, that sort of thing, in the context of just massive amounts of data, provide amazing opportunities to gain insights that, we might not otherwise have. I do worry about, the pitfalls when we assume that data is good data, because, the devil's in the details. And, I remember back, 15 years ago when I think companies would say, buy a pot of lupus samples, let's get blood samples from 100 lupus patients, and they would view those as lupus samples. Well, we know that there's so much difference from one patient to another, from one healthy person to another, and you don't really know how to analyze that data in an effective way unless you really take account of the individuality, both genetically, biologically, and then everything we do to the patients. What drugs do we give them? So I guess I'm optimistic or curious about what's going to come out of sophisticated analytic approaches to all of this big data. But at the same time, I'm a little cautious about believing what comes out immediately. I get very excited by trying to link, and this is what I said earlier, really, link very basic science observations coming from other fields to study of lupus patients. And I think each helps the other. So we get deeper and deeper into understanding the way cells work, how they signal, how they regulate themselves. And I find all of that very fascinating. And sometimes it's informative with regard to lupus. So all of that is what I find exciting. What's promising, I do think are the advances being made in lupus clinical trials. And the lupus field has struggled so much over the last 20 years to find ways of having successful clinical trials. And I think a lot of that is just, related to all of this heterogeneity that we always talk about. But I think it's mostly just a learning process. We had to learn that you couldn't treat patients with high doses of steroids for too long in the context of a trial, or you will blunt any signal that you might otherwise be seeing. So I think there's been great experience. And now there are, I think, a number of examples of successes in trials with drugs targeting very, very different mechanisms.
Do you think lupus is a disease? Because we talk about the heterogeneity. So is it really a disease, or are there going to be some patients who are interferonopathy patients and some patients who are hypocomplementemic patients? Mm-hmm. I mean, are we learning by calling it a disease, or are we impairing our ability to treat people because we're putting them in a bucket?
I think that, we can all debate this. And I think fundamentally it's a disease, but any individual can get to that disease in any number of ways. So maybe that's a compromise answer. So I guess,
a common pathway to a lot of challenges.
Yeah, I think that type 1 interferon is pretty fundamental to lupus, and you can probably get there without activating the interferon pathway, but that's rare. And I think the other feature, though, of lupus is the capacity to develop autoantibodies that target nucleic acid binding particles. So if you think about the genetic so-called interferonopathies, like Aicardi-Gouttières syndrome, or some of the other syndromes of children that involve, mutations in certain signaling pathways that end up driving interferon, their clinical pictures are not necessarily lupus. And they might have low levels of antibodies, but they rarely would have the kinds of antibodies or at the level that we see in lupus patients. So what's missing there? I think what's missing is the production of autoantibodies. So why does that happen? Well, I think it has a lot to do with genetic risk, mostly HLA related, or in part, let's say HLA related there. But I don't think we understand either of those two components, the interferon pathway activation or the generation of autoantibodies. And that's where I get very psyched up to try to figure stuff out. Why would an individual drive, one or the other or both of those pathways? But you brought up the issue of complement deficiencies. I mean, that might be an example of a very discrete genetic alteration that does head you toward lupus, but in a very specific way, but that's very, very, very rare and not the common.
No, but they're hypomorphs, and there are lots of individuals who aren't deficient, but are dysfunctional that may drive either autoimmunity or their end organ disease. And kind of in a sense, you're talking about this end pathway where you look similar, even though your triggers are different. And then the issue that you raise in terms of why you make the autoantibodies, what are the environmental triggers? And we really don't understand that, I think, at all.
And we haven't mentioned the female skewing issue, which, we don't talk about it that much because we don't have the answer to it. Although I think maybe the field is getting closer with the focus on the X chromosome and something about the X chromosome that maybe impacts toll-like receptor pathways. We'll see.
And neutrophils. And neutrophils, maybe because of toll-like receptor pathways. But it is interesting that we're not talking about that because it's hard. And we don't know.
I'll ask you one more question and then we should stop. Okay. So if you come to some kind of conclusion with your pregnancy project, anti-TNF agents are helpful, and now that has been implemented in practice, and you have a predictor of pregnancy outcomes. What will you do next? Or, would you ever go off in a completely different research direction?
So I don't think I have an infinite time. So the first thing I would do is I would drink a lot of wine because I'd be happy. But after I finish drinking wine, what I'd want to do is kind of my dream, and it's been something I talk about it a lot, is to say that lupus is a window of amplified, accelerated phenotypes. And if we understand disease mechanism through this window of lupus, we can apply it to the broader population. And, when I tell people, I work on lupus, they're like, well, what's that? Or that's like this weird disease that women get. What I really want to do is do a trial to prevent preeclampsia in non-autoimmune patients, because all of our mouse models and some human observational studies suggest that similar pathways play a role. It's just the frequency of these terrible outcomes is lower in the general population. It's enriched in lupus, so you can study it. So my goal is to do a gigantic national study of women at high risk for preeclampsia, and we can do risk stratification reasonably well, but still the numbers are going to be, 5 or 10%, or maybe close to 20. But in our current trial, the numbers of background should be 44. We'll never get 44. So my dream is to use my therapeutic, whichever one it ends up being, in the general population and impacting on preeclampsia, and arguing that studying unusual diseases that are exciting impact the whole world. And I know it's grandiose, but we should support all kinds of ideas that ultimately, that you don't really think matter in a big way, but ultimately can have tremendous impact. So that's my dream, to do that trial.
Well,
so I'm not deviating. I'm still like going straight, looking at the same questions because it's me and it's my wiring.
Yeah. So I guess if I answer that question, I'll also be very, very grandiose, maybe more so. And to some extent, I'm not really serious in saying this, but, I think what I'd like to really understand is what makes humans human, with regard to how genomes developed over evolutionary time, what happened that allowed brains to develop in this way with the complexity and the flexibility and, the very complex wiring that's allowed us to be what we are. So I imagine I'll never quite get to that point, but I am very curious.
Traveler. You could be a time traveler, take your science, but move back and see the steps. That's exciting. That's bold, Peggy. You're bold and creative and fearless.